Okay. So what are we doing in class today? It's more genetic mapping. The goal is, which we started this process last time, for you to be able to distinguish what it means, or to determine what it means when two genes are 50 centimorgans apart. How do you interpret that piece of information? I don't want everybody to be able to do three-point mapping as it's been described in the chapter, but I want everybody to be comfortable with why the process of three-point mapping is necessary. First, what it is, and second, why is it necessary? And then at the end of class, we're going to talk about why linkage, having the same gene near each other on the same chromosome, is important with a specific example from humans. Aside from not doing three-point test crosses, I also don't want you to worry about the concept that you read about for today of genetic interference. If I had been nicer and been thinking about it, I would have told you before you read the chapter, I apologize, but we're not going to talk at all about genetic interference. So you're not responsible for that content. So sometime during the last class, I asked everybody, which most of you did, to submit a drawing of a physical map representing the locations of two unlinked genes. This is just a smattering of all of the submissions that I received from you. So I've numbered them all so that we can discuss them a little bit more easily. You might recognize one of these as your own. You don't have to claim yours if you don't want to. But I'd like everybody to take a look at these for a minute. Talk to a neighbor if you want to. I want to know if you have any questions about whether or not all of these are accurate, correct answers to the task. Do all of these pictures represent the locations of two unlinked genes? So think about that a minute, maybe a minute and a half. Talk to a friend. Which of these don't meet the criteria that I requested? Yeah. This is not the point. Just for practice. Wow.
No way.
Okay. So at this point, what do you think? If there's one of these examples that your group or you individually want to talk about, which one is it? What number? Number 3? Okay, so let's start with number 3. I like number 3, which is why I put it up here. Thank you for participating. I like all of these responses, which is why I chose them out of all of the responses. This random sampling, but there are some qualities to all of these. Number 3 is one that we're going to discuss. Right? One of you or more of you, more than one group suggested, let's talk about group 3, or number 3 here. What's the issue? So we've got two different alternatives, 3 on the left side and 3 on the right side. There are two different drawings here for submission number 3. Which one shows genes A and B that are unlinked? The left side or the right side? Say it louder. The left side. Right. Gene A, 2 copies, so this individual that was obviously thinking about a diploid, we got 2 copies of chromosome 1, let's say with gene A, 2 copies of chromosome 2, say with gene B. They're unlinked, they're on separate chromosomes. What about the drawing on the right for number 3? Why does that not meet the criteria that I requested? Because they're on the same chromosome. According to our definition of linkage, 2 genes are on the same physical piece of DNA. They are linked. But I really, really like this response. It's the same as in number 4. It's different drawings, different students, but the same concept. Students that drew and submitted 2 genes on 2 separate chromosomes, that's unlinked. But then both of these students, and a lot more, not just these 2, also drew in the exact same submission to me that second drawing. Like this one. Right there, and this one. 2 linked genes. Why did these students submit that version of the drawing to me? What happens when 2 genes are on the ends, as drawn here, of the same chromosome? How many centimorgans are going to be separating those 2 genes? 50 centimorgans. And that's what we're going to talk about in class today. That 2 genes, if they're on the farthest reaches of the same chromosome, will have the exact same map distance as 2 genes that are on separate chromosomes. So these students, I think, if I can interpret your artwork for you, we're trying to point out that when genes are 50 centimorgans apart, they could either be on the opposite ends of the same chromosome, they could be on separate chromosomes. The point I was trying to make with this is that if genes are linked, which it said in the question, they're on the same chromosome. If they are unlinked, they're on separate chromosomes. Any questions or concerns about this point? So linked genes are on the same chromosome. If they're unlinked, they are definitely on separate chromosomes. So the answer to the next question is yes. Linked genes can be 50 centimorgans apart.
Dr. Rock?
Yeah.
I might be remembering this incorrectly, but did you say that when the 2 genes are on top of each other during crossing over, they're right on top of each other, have no probability
Right.
being
Separated.
separated or being linked?
Right.
But if they're a little bit apart from each other, then they have a possibility of being linked?
Right. So it's another way to review this. Here are genes A and B on the same piece of DNA, same chromosome. They're just different physical distances apart. On the left side, the 2 genes are right next to each other on the chromosome. In the middle example, they're some distance apart. On the third example, they're at the opposite ends of the chromosome. You should be able to tell me what the map distances are. At least by the end of today, you should be. So on the left side here, how many centimorgans apart will those 2 genes be? We're using the number of crossover events as a metric for how far apart genes are.
0.
So if there's no chance that recombination happens between genes A and B, their genetic distance will be 0. Pick your favorite map unit. Recombination fraction, centimorgans, map units. I'll just say recombination fraction for that example. On the right, which we just talked about, if genes are at the opposite ends of the chromosomes, what's the probability that a recombination event will happen between them during meiosis? There has to be 1 crossover per chromosome in every meiosis. So if you have to put a crossover event somewhere here between, on this chromosome, where is it going to happen? Between, it has to be. If A and B are at the ends of the chromosomes and 1 recombination event has to happen, It's going to happen between them. That's independent assortment of linked genes. That's 50 centimorgans. 50% of the gametes produced from this will have recombinant. Okay, and then the example in the middle is somewhere between 0 and 50 centimorgans. There's some probability that a recombination event lands in between the two genes. There's some probability that recombination will happen not separating the two. So their genetic distance will be somewhere between 0 and 50. So those are the extremes and everything else in between in the spectrum of genetic distances. Let's play Prove the Professor Wrong. So what's the problem with what I drew here? Especially for those of you that didn't hear the question. What's the range of recombination fraction? It's not 0 to 50. It's 0 to 0.5. If I wanted to put something in here that was accurate, what would I replace RF with? MU or centimorgans. So here I had automatically in my brain, because I do this, converted recombination fraction to map units or centimorgans would be 0 to 50. Recombination fraction would be 0 to 0.5. Thank you for catching that. So this is just a brief reminder of what we talked about last class. So now we're getting into a little bit of the 3-point test cross. The question we're trying to ask in this part of class is, how do you tell the order of genes along a chromosome? Here we looked at genetic map distance between genes A and B, and we calculated that they were 10 centimorgans apart. That was last class. Last class we also looked at genes A and D, and we calculated that they were 5 centimorgans apart. So we're talking about gene A, gene B, gene D. Go ahead.
I have a question about the couple slides before. For
Yep.
ours would be between 0 and 0.5 since the centimorgans are
0 to 50.
0. Okay, but when you put it in RF?
I'm sorry. And then at the end of class last time, I asked everybody to try this. Draw a physical map, the chromosomes. Tell me where A, B, and D are located. Just like the drawings that were submitted for last class, Google assignment. Physical map, where are the genes relative to each other? How many different versions of a map can you draw? So we've got where are you going to put genes A, B, and D? Those of you that tried this. Are they all on the same chromosome? Genes A and B, as it shows up here, we concluded that they were 10 centimorgans apart. Genes A and D are 5 centimorgans apart. Are all 3 genes on the same chromosome? What's the genetic distance that you have to have between 2 genes if they're on the same chromosome? What are the range of possible genetic distances between 2 linked genes? 0 to, what did you say? 0 to 50. Well, what happens when it's 50 centimorgans? Where are the genes necessarily if you say that there are 50 centimorgans apart? They're either at the ends or they're on separate chromosomes. So if A and B are 10 centimorgans apart, are they on the same chromosome or are they on different chromosomes? They're on the same chromosome. The only way you get genes that are physically unlinked is to have a genetic distance of 50. So A and B must be on the same chromosome. A and D must be on the same chromosome. Their genetic distances are less than 50. So, there's a chromosome. There's A. This is just arbitrary at this point. I'm just saying A exists here. Now what do? I put B somewhere that's 10 units away. Okay. Now is where you have to think carefully. Where am I going to put the third gene? Where does D go? So D could go in the middle. So D could go there. That would make it 5 centimorgans from A, which we calculated. Or it could go on the left side of A. Right? Either place we put D, it's 5 centimorgans from A, as the map calculation suggests. So it's either DAB or it's ADB. So we haven't answered the question yet. What else do we need to answer this question? What other piece of information could we identify that would help us figure out what order the 3 genes are along the chromosome? Distance between B and D. Right? If we're dealing with 3 genes, there are 3 different pairs of distances between those 3 genes. There's A to B, A to D, and B to D. So what do we predict if D is on the left side of A? What's the D to B genetic distance going to be if we calculate it? It should be 15, because what you should have read in the chapter I haven't mentioned in class yet, but now I'm going to, genetic distances are additive. So if you have 2 adjacent genetic intervals, genetic distances, 5 and 10 centimorgans, the total distance should be 15 centimorgans. Okay. What will it be if D is in between A and B? If D is there, what's the D to B distance going to be? The 10 minus the 5 that's there, so it should be 5. So we're either going to find that B to D is 15 centimorgans or B to D is 5 centimorgans. That answer is going to tell us which of these 2 alternatives is true. Okay. Now's your chance. So here's a cross between some parents with B's and a parents with D's. So go ahead and please calculate the genetic distance. Talk to a friend if you want, of course, absolutely. Do about 5 minutes for this, and I'm going to open up a single question in Socrative. This is a type of Socrative you haven't seen before, but when you're done calculating it, please enter the map distance into Socrative. Okay, let's see where everybody's at right now. I've seen some submissions to Socrative, so I know some of you have gotten through calculating it. Getting a wide variety of answers, which is good. It means there's still a little bit of discussion to be had. So what's the first step we need to do to calculate genetic distance? What was the first thing you did? So what about the 2 parents? So you cross the 2 parents, that produces what's this genotype that is in the F1 generation? It's a big B and a little b and a big D and a little d, which is a heterozygote at 2 loci, which makes it a dihybrid. Okay. And what sort of a cross do we do when we're doing genetic mapping? It's a dihybrid test cross. So we need to put the tester individual genotype here. So in this example, what's a tester genotype? It's homozygous recessive. So it's homozygous for the little alleles, the lowercase alleles, that is. Okay. What else do we need to do to figure this out? What are the gametes produced by the dihybrid, by the tester? Right. So we know that those little b's and little d's came from the tester. What's left tells us about the types of gametes that the F1 dihybrid produced. That's why we use a tester, because we know which alleles it has. It's got the recessive alleles. That's how we know we can cross out a little b and a little d from every F2 genotype. What's the last thing we need to know before we actually do the calculation? Can't assume. You might have assumed this today. You can't assume this. What's the calculation of genetic distance based on? What sort of measurement is it?
The recombinant versus the parental.
Right. It's the number of recombination events that happen between two genes. So we need to know which of these combinations are parental and which are recombinant. Which of those are evidence of crossing over happening during meiosis? So in order to figure out which are parental and which are recombinant, where do we look?
The haplotypes of the G0.
Yeah, we have to look at the parental generation. So what haplotype did that P0 parent produce? Big B with little d. And we've got little b's with big D's. So of those four F2 haplotypes, what's the top one? Big B with big D. Parental or recombinant? It's recombinant. It's not a haplotype. Big B with big D was not up here in the P0 generation. So that is a recombinant type. Little b with little d is also not present in the P0 gametes, so it's recombinant. And then we've got parental. So what's the genetic distance? It's the two recombinant types. There are 69 big B, big D recombinant gametes. There were 81 recombinants of the other type. There were 1,000 total offspring. So the recombination fraction is it 50 or is it 15 or is it 5? It's 150 over 1,000, or a recombination fraction of 0.15, or multiply that by 100 and you get 15 centimorgans. So now we know the actual order of the three genes along the chromosome. It has to be the B and D, which have the farthest genetic distance we calculated, 15 centimorgans, have to be the farthest apart genes. What's in between them then? What gene? A has to be in between. So important point here is that you have to, every time, look up here at the parental genotypes. You have to know the parental genotypes and know the gamete haplotypes that they produced to know which haplotypes are recombinant and which are parental. I tried to trick you, I don't know if I did. These are different genotypes than I used in crosses that we looked at last time. Last class, what did I do? The two crosses we looked at, what were the genotypes? One parent had all capital letters, and we crossed that in both of the cases we looked at last class, with the other parent having both lowercase letters. Now I want you to be careful of just assuming that when you see two capital letters together, two lowercase letters together, that those are the parental types. That's not always the case. You have to figure it out from looking at the genotypes of the parental individuals. Yeah.
Can you just, repeat again, what you did, the A was in the middle? What's
Okay.
100 over, 150 over 1,000 type?
So 150 over 1,000 gives us the map length between A and D is 15. So if A to D is 15, thank you for asking me to do this explicitly. So if A to D is 15 centimorgans, sorry, B to D, never trust a geneticist, absent-minded. We also calculated that A to D was 5, this was last class, 5 centimorgans, and A to B was 10 centimorgans. So where are we going to put gene A on this map to make all of these data true? Where can we put A so that it's 10 centimorgans from B and 5 centimorgans from D? And 5 plus 10 equals 15. That's one bad genetic map. Bad in a good way. You know, I'm hip. Okay. So what are the take-home messages from this example? Right. If you want to know the order of genes along a chromosome, and if you have no DNA sequence information for you to determine that just by looking at the DNA sequence of a chromosome, which is still true for most of the species on the planet, we don't have any DNA sequence in the database for them, the largest genetic distance you calculate will be the one that's separating the farthest apart genes, and the third gene will usually be in the middle. And to do this, this is where the trihybrid cross that you read about in the chapter comes into play. To deduce the order of 3 genes, you either do 1 cross with 3 genes. That looks like this, which is why we're not doing that, because it's just an order of magnitude more complicated. You deal with 3 genes at once. This is what a trihybrid test cross would look like. You can do 1 cross and get all the information, or you can do 3 separate dihybrid crosses. You do a cross that involves A and B, A and C, and B and C, 3 dihybrid crosses, you get exactly the same information. The genetic distances between all of the 2 possible combinations of 3 genes. So you don't have to do a trihybrid test cross, it's just less work in the laboratory. You do 1 cross instead of 3. So if I tell you that genetic distances are less than 50 centimorgans, what's the conclusion? They're linked. Yes, they're on the same chromosome. If you discover that genes are 50 centimorgans apart, what's the answer? Are they linked or are they unlinked? We don't know. Right? Genes that are 50 centimorgans apart are either at the ends of the same chromosome, they're linked, or they're on different chromosomes, they're unlinked. So you can't give an answer if you calculate 50 centimorgans. You don't have enough information yet. So if you ran into that situation, what sort of additional information would you want to let you answer the question of are the genes linked or not linked? What does the geneticist do? What did we use in the last example to figure out the order of 3 genes or the relative locations of 3 genes? Oh, I gave it away. What do we need? We need a third gene. So for example, let's say you measured the genetic distance first. You measured the genetic distance between Q and R. What is their distance in this case? 50 centimorgans. And you wanted to know, are they on the same chromosome or are they on different chromosomes? How does the presence of gene T, in this case, help? So say you also discover this third gene, gene T, and you calculate its genetic distance to Q. You discover that it's 50 centimorgans. So at this point, you've got two possibilities. You've got Q on one chromosome, you've got R on one chromosome, you've got T on one chromosome. They're all unlinked. Or the version that was originally drawn on the screen. What's the difference between these two versions of this? This is all consistent with the data we have so far. Q and R are 50 centimorgans apart, Q and T are 50 centimorgans apart. What are we going to discover about R and T when we measure their distance? Less than 50 centimorgans. So if R and T are less than 50 centimorgans apart, we conclude that they're linked. And they're both separately 50 centimorgans apart from gene Q. So that tells us that Q and R, the original ones we had a question about, we calculated 50 centimorgans, are they linked or are they not? If R is linked to T, and T and R are both 50 centimorgans from Q, the only possible outcome is that R and T are linked and the Q is on the second chromosome. We'll do another practice at some point with this concept because it's a bit funny. Good qualitative analysis question to ask on an exam. So you'll usually need a third gene to determine whether or not a calculation of 50 centimorgans means that genes are linked or unlinked. So one more piece of information. Any questions at this point? Besides, why am I putting a picture of an axe-wielding hitchhiker on the screen? We're going to talk about genetic hitchhiking. There are no other. This is the example of why linkage can be bad for humans. There are reasons that linkage can be beneficial. Sometimes it's really useful to have genes that are right next to each other on the same chromosome. But in these examples, oh, there it is. Crohn's disease, diarrhea, and other symptoms has something to do with linkage in humans. So Crohn's disease is this debilitating disease that a lot of people have around the world. And fortunately, I claim that I do not suffer from Crohn's disease. But it's on the rise. So there's an increase over the last few centuries in the number of people that have Crohn's disease. And it turns out this is a gene or a disease that has a genetic basis. So that's the disease, Crohn's disease. It makes you sit on the toilet for a long time and have stomach pain and diarrhea and bloating. And for a long time, it's been known for a while now, that there are different regions of our different chromosomes that contribute to your probability of developing Crohn's disease. It's a genetic disorder, or at least partly genetic. And this is a drawing of part of human chromosome 5. Right? We've got an ideogram of all of human chromosome 5 here at the top. And what I've done here is blown up the tiny little slice of chromosome 5 that's shown by that red bar on the ideogram. So this part of chromosome 5 is a just tiny fraction of the total human chromosome 5. And in this region, there's a gene that's responsible, or at least partly responsible, for lengthy toilet sitting, the phenotype. So there are only a handful of genes in this tiny little region. There's a gene called OCTN1, a dietary antioxidant transporter. It means it takes the good things in chocolate and wine and so forth from your intestine into your bloodstream for you to use in fighting the free radicals. There's interferon regulatory factor, IRF1, and there's an interleukin gene. And there are a couple other genes in here. We'll focus on these three, though. It doesn't matter necessarily what they do or if you know what they do. Let's just take a quick vote. How many of you think that the dietary antioxidant transporter is going to be the gene that causes Crohn's disease? See, just a show of hands. One student. 2, 3, 4, okay, bandwagon, a little bit of picking up. What about interferon regulatory factor? Anybody? About the same number of students. Nobody wants to vote. So it must be interleukin then, all of you that didn't raise your hands. Right? Oh, come on. It's got to... No? Seriously? Hump day and you won't even raise your hand. So nobody wants to hazard a guess, essentially. We had about 5 students raising their hands for each of these 3 genes. We don't know what the genes do. One of them is the cause. Now, it turns out it's OCTN1. So for people that have Crohn's disease, they all, or mostly all, share one version of the OCTN1 gene, an allele. This allele, though, what does it do? Is it good or is it bad for us? What does this allele do? This is like the group question on the exam. Read the abstract and tell me what it does. What's the outcome of having, if your chromosome has this version of OCTN1, is it good or is it bad for you? Increases dietary antioxidant absorbance. Does that sound good or bad? People, they've been telling us for years to drink more red wine and eat the chocolate and eat the rainbow of fruits and vegetables, right? We need our antioxidants. Fight aging. So it turns out that this OCTN1 allele is beneficial to us. The people that have this take up more antioxidants from what they're eating into their bloodstream, and maybe they live longer. Who knows? So it's probably not OCTN1. There's a relationship between the people that get Crohn's disease and having this one specific version of OCTN1. So what they did next, these researchers, Huff et al., this is published in 2011, they looked, in each of these columns is a different gene in this region of chromosome 5. So we've got P4HA2, PD-LIM4, there's OCTN1, OCTN2, interferon regulatory factor, and interleukin 5, left to right. All they wanted to know, and all this graph shows, is for each of those genes, they want to know which gene is causing Crohn's disease. So they have a hypothesis that if they measure how much of that gene is transcribed into messenger RNA, that will have some relationship with whether or not individuals develop Crohn's disease. So if we look here in the first column, we've got a group of people that have Crohn's disease, and on the y-axis is messenger RNA expression. That is, how much messenger RNA is produced by that gene, by those individuals that have Crohn's disease. Okay. And then we compare that next door to control individuals, people that don't have Crohn's disease. Is there any difference in the amount of that gene that's produced by either population? Or are they the same? Each of these points represents the amount of gene that's turned into messenger RNA. So is there any difference between Crohn's and control? But the points basically are in the same y-axis range. There's no difference. So that rules out that gene as being likely contributing to Crohn's disease. What we're looking for is the difference between the people that have Crohn's disease and the control people, the people that don't have Crohn's disease. Where do we find that? Where do we see a difference for one gene between the Crohn's people and the control no Crohn's groups? Where's the biggest difference? IRF1. So here's the story. This is the punchline. The people that get Crohn's disease, the geneticists initially saw, have this beneficial mutation. The asterisk in black, that's the one that increases dietary antioxidant absorbance. However, those same people that take up more antioxidants from their gut, also on the exact same chromosome, just a little bit farther down, in a linked gene, in IL5, there's a mutation, IRF1, that increases the probability of getting Crohn's disease. So the basic question is, what happens when on the same chromosome, chromosome 5 in humans, you have a good allele and a bad allele next to each other on the chromosome? One increases antioxidant absorbance, one gives you Crohn's disease. They're linked. The good allele, the bad allele, two different genes. They're on the same piece of DNA. They're inherited together. So what do you think happens? So imagine you have a case like this. We've got chromosome 5, a horizontal black line, and these are chromosome 5 from 5 different individuals, let's say. So we're looking at chromosome 5 from 5 different people. Okay. Which of these chromosomes is the one that you do not want to have? Chromosome 1, chromosome 2, 3, 4, or 5. Which is the worst of those 5 versions to have? Number 2, right? It's got a detrimental mutation, gives you Crohn's disease, and it doesn't have that beneficial mutation. So this is the worst possible chromosome to have. So imagine that these are 5 chromosomes from 5 different people in the human population as a whole generations ago, a long time ago. What's this same screen going to look like after time passes? Which of these chromosomes are going to no longer exist when we fast forward through time? Chromosome 2. It's the worst chromosome you could have. So we expect not to see chromosome exist anymore when we fast forward through time. So go several generations into the future, what does the human population look like? We've got a bunch of individuals that have chromosome 5 with the good and the bad linked alleles on their chromosome. And we've got a bunch of people that don't have either. And I should point out that this assumes that there's a net benefit. That is, that the sum of how bad having the bad allele is and how good having the good allele is in total gives you an advantage, gives you a benefit. So it's better to have both the good and the bad together than not to have either. So if we put numbers on this, we could say that detrimental gives you -2 hit points and beneficial gives you +3. So in sum, you're better off having the good and the bad allele on the same chromosome rather than having neither. Right, so this is, you get a 0 and a 0, so your score for having neither is 0, but you've got an advantage +1 if you've got both. So what does the human population look like today? Most people have the combination of the antioxidant transporter higher efficiency version and the propensity for getting Crohn's disease. This is just because of linkage. Together, those two alleles are better than having neither. So the beneficial one is expanding. It's passing more from person to person, generation to generation in the population because it gives their offspring benefits. Now we're talking about evolution. So why does the detrimental allele stick around? Why do we still have it? Because it's linked. What would we have to do to get rid of the detrimental allele at this related gene, the nearby gene? There would have to be crossing over between those two genes. How close are they? The beneficial and the detrimental alleles of these two genes, as it says up at the top there, they're separated by about 100,000 base pairs on chromosome 5. That's a lot of DNA. 100,000 base pairs. Except, what does it say underneath that? Chromosome 5 is how long? 180 million base pairs long. So when you think about every meiosis, recombination has to happen on chromosome 5. Recombination is going to happen somewhere in that 180 million base pairs of DNA. What's the probability that it happens in the 100,000 base pairs between these two genes? It's really, 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 really low. So we just haven't been able to physically separate the good and the bad alleles. That's linkage. In fact, that's genetic hitchhiking. Get the analogy? It's like the bad allele is the hitchhiker, the guy with the hatchet. Right? Some good person comes along, going somewhere, like going to fixation in the population because that person's beneficial. They are bringing the hitchhiker with the axe along for the ride because they're genetic hitchhiking. So one beneficial allele goes to more and more people in the population because it's beneficial. it drags along with it everything that's linked to it, bad or not. Because the net is benefit. Everything else that's linked comes along for the ride. On the chromosome, that is. So, we've got a reading assignment for next class, and I'd also like you to watch this movie, which is a great introduction. It's short, it's a couple minutes. It's a great, I didn't make it. It's a great introduction to what we're going to be starting to talk about for the next couple of chapters. But please remember, if you didn't get your exam, email me. I will resend the email, okay? We've got office hours now if you want to come chat.